Let's review some key concepts of the 20 amino acid lecture. The enantiomer, as we learned in the chemistry, this is the chiral molecule to a non-superimposable mirror image. So it's like our hand, our left hand and right hand, they're mirror image, but they can stack each other and totally overlap. And optical activity, that's not that important in this class. This is the focus on the organic chemistry. And as we need to know is the L isomer is only appear in the cell. So we only see the L isomer because there are more steric strand or something else in the D isomer. And the hydrophobic interaction, this is the non-polar molecule that interact with each other, so they have long hydrophobic hydrocarbon chain. They only contain hydrogen and carbon, and maybe they have the benzene ring or something, and this is called the aliphatic. And disulfide bond, this is the important things in this lecture. It is a covalent bond that link two cysteine together to form the cysteine. And the cysteine is the only amino acid that can form the disulfide bond. We can see there is a thiol in the cysteine and the methionine, but methionine, they can form the disulfide bond, so we need to know. And there's a little tip to remember, the Lioger, that means you lose electron, that is an oxidation reaction. If you gain electron, that is a reduction reaction. So from 2cysteine to cysteine, it is lose electron and form the disulfide bond. So it will lose electron, and this is an oxidation reaction. If you want one cysteine to form one cysteine to form two cysteine, you're gonna gain electron and break the disulfide bond, and this is a reduction reaction. And why we need this disulfide bond means this can stabilize the protein structure. And there is a protein disulfide isomerase. You can see the ASE at the end. So this means this is an enzyme that helps the oxidation reaction. And there's another way. If you want to break the disulfide bond, you want to form two cysteine, you want to use the BME, the beta mercaptoethanol. So this is a reducing agent. And there's secreted protein and cytosolic protein. Disulfide bond formation is the post-translational modification when it passes through the endoplasmic reticulum. And let's take a quick example of the hormone. Hormone is one of the secreted protein that insulin that's secreted from the pancreas will float around in the bloodstream, and they can form disulfide bonds, so they can maintain their structure. And for the cytosolic protein, they contain free cysteine because they are reducing nature of the cytosol. Because so in the cytosol we don't have disulfide bond because there's reducing molecule of the cytosol, so the cysteine cannot form cysteine. And in the lab we have the BME I mentioned earlier. This can break the disulfide bond. And we know there's perm, that means you want your hair to be curl or something. And we have alpha keratin in our hair, and this is a type of structural protein. And then this means you need to break your disulfide bond and you change to other shape you like, and then you form the disulfide bond again, and this is what the perm look like. And the straightener, that means our hair, there is hydrogen bond in them, between them. So we can use the heat to break this non-covalent bond, such as hydrogen bond. And if we wash the hair, the hydrogen bond will unfold, and and your hair will look what your original like. And post-translational modification, disulfide bond is part of them. Covalently modification means you add some group on your protein or something. Phosphorylation, that means you add a phosphate group. Ubiquitination, that means it is a marker for the apoptosis. And glycosylation, that means you add a sugar. And there are other type of little functional group add on them, the acetate, methyl, hydroxyl, or carboxylic acid. And the post-translational modification can be also amino acid modification. They can add or remove functional group to form other amino acid derivative. Like, we can take an example of the asparagine and glutamate. They can form the aspartate and glutamate because the aspartate and glutamate, when they add on a amino group, they can become asparagine and glutamate. So asparagine and glutamate, they're not the essential amino acid, because we can do a simple step to form them. I mentioned this. And there's homology search. We know the central dogma of the biology, that's DNA transcribed to RNA and translate to the protein. If we have a mutation in our primary sequence, that means our amino acid, there's 3 types of response or effect. The first one is the silent. That means there's no effect on the protein. If you see the table, you can see something like UCC. I don't remember the three code. That means let's take an example like UCC and UGG. They all can become tryptophan. Let's just take an example of that. So there's no effect. And non-conservative means that change all about the amino acid structure and function. And conservative means you conserve some of the structure or some of the property of the amino acid, because maybe you have the same functional group like leucine and isoleucine. And if you want to test the system from how form the disulfide bond, maybe you can thiol and hydroxyl, they can also both form the hydrogen bond. Maybe that's not the thiol work, maybe there's the hydrogen bond. That's not the focus.